All right, so hi everybody. I'm Eric. I know all of you. And so I am in the molecular medicine program, and I'm doing a project with Michelle Ferguson in the pediatric oncology department, and we're looking at Langerhans cell histiocytosis. And I think some of you were in my last talk, but some of you weren't, so I apologize if you see some reused slides, but I want to make sure we all kind of had a basic understanding of what this disease that I'm talking about is, because it's kind of one you don't really hear about everywhere. So I think just starting with, I think this is kind of my big project as a whole. My last talk I did maybe 4 weeks ago, I talked a little bit about aim number 1. So today I just kind of keep it interesting for myself and talk a little bit more about my second aim, which is kind of more immunology based. I apologize for you non-basic science people. It's going to get very basic sciencey in a little bit. So I guess just to really start, I kind of want to breeze through kind of what this disease is, and I think historically speaking, it was just marked by a bunch of different names because the disease has a really kind of wide variable presentation. So before, we had all the fancy diagnostic tools. Physicians were just recognizing different patterns of disease and calling them the same thing. And then later on, once we got more specific ways to look at it, both on an electron microscope, and then finally now we use different staining techniques. We realized that actually this was kind of the same disease, even though it had such diverse presentations. And so today, as we understand it today, we think of Langerhans cell histiocytosis as this accumulation of what is termed Langerhans cell-like cells types. And then today, historically, and even today the gold standard is still electron microscopy to look for virulent granules, but today oftentimes they'll do staining. This is the CD1A and CD207, which are just different surface stains that they use to identify these pathologic cell types. Interestingly, this disease can present at any age. And we've seen there are cases in neonatal, and as well as in older populations. It is more common in children, and thus it's considered a pediatric oncology disease. And their estimated rates are about 2.5 to 5 cases per million in the pediatric population, so a fairly rare disease. Interesting enough too, and kind of what got me kind of interested is that in terms of different presentations, we see this really severe multi-system disease that tends to be more common in the pediatric population as well, for reasons we don't know. So I think there are two big different classifications we think about. There's a single system, LCH, and then as well as multi-system. And the multi-system, of course, is a really bad actor here, because you get, and again, this multi-system is also split into low risk versus high risk depending on which organs are involved. So high risk organs will be liver, spleen, and hematopoietic system, which is not surprising. That might not be a good thing to have affected. And this, again, like I said, is higher in pediatric population, and actually really young kids as well, under 2 years. And here again, which is just the percentage of what you see if you look at everybody with Langerhans cell histiocytosis. So the most common is not this multi-system, but it tends to be more of a single system disease, with bone being really the most common. But again, as you start moving down here, you start getting to more and more bad clinical presentations. Treatment, I don't want to talk about too much about it. Single system tends to be local. Solitary bone lesions really respond well to just simple curettage. But multi-system, again, is going to need systemic chemotherapy, along with all the morbidity that's associated with that. Survival curves. So this, I should mention that this goes up until 1998, so chemotherapy has gotten more better. It's gotten more therapeutic. So we shift these lines up a little bit, but I just want to put this up here that here we have the single system, and then here we have this really bad multi-system, and see big drop in difference. And I think this is kind of what got me interested in this project was what separates this from this disease. If it's the same cell, why do some kids just come in with this, and why do some end up down here? So I think before we go a little farther, I'm going to talk a little bit about kind of like the basic biology behind all this. So the Langerhans cells are the dendritic cells of the epithelial mucosa. Like I said, they do have CD1A, and CD207. These are CD1A, CD207 positive cells. That's how they are defined histologically. Interestingly enough, I mean, we don't really know a lot about them, which is kind of interesting. We do think that they tend to hang out for quite a while in the skin, and they have really long half-lives, and they seem to kind of renew by local proliferation. But other than that, not too much is known about biology. It's kind of an interesting field. They've looked at it in mouse models. They seem to come really, really early in development. Here, they migrate in utero, and then they kind of differentiate later after the birth. And again, this is mice. We don't know if this is true in humans. So that, again, is also kind of an interesting point about these kind of LC populations. Like, are these just residents that take up shop really early and then don't seem to move? In terms of where they develop from, so these are part of the hematopoietic system. They are different from the lymphoid. Here's the lymphoid go down to B and T cells. Here we have myeloid precursors, and I must say, like, this to this is really not well known on this kind of. They have found some precursors again in mouse models, but in humans it really seems to be a dearth of kind of what is the progression in terms of getting from way up here in the bone marrow all the way down here in the skin. Interestingly enough, though, this they have, in terms of bone marrow transplant patients, this does seem to be something that comes from the bone marrow or at least circulating the blood, because it does seem to repopulate post bone marrow transplant as well. So these precursors are able to populate the skin again if needed. So I think recently a group did some comparative gene expression arrays on Langerhans lysocytosis. And what they were doing, they were comparing the LCH cells, CD207, cells in the LCH lesions. These are thought to be the LCH cells. And then compared it to normal dendritic cells in the epithelium. And they compared kind of their gene expression using array-based technology. And what they actually saw, that they had a bunch of, you compare kind of what one cell is expressing versus the other. What they saw was that the genes that the LCH cells were expressing seemed to group more closely and kind of resemble more of an immature myeloid phenotype than actually a Langerhans cell itself. So what they proposed then, that this LCH, or this pathological cell that looks like a Langerhans cell, may not actually come from a Langerhans cell itself, but may come from higher up in that developmental tree. So, instead of coming from here, they thought maybe it's, up here somewhere, where we don't really have a good sense of what exactly is going on up there. And so I think in terms of, my project and looking at, now that we know that maybe this isn't from an LC cell, maybe this is from a myeloid precursor, but really what I'm interested in is, like, what is driving this development to this pathological state. I think there are some big barriers in this disease, and that, this is a rare disease first and foremost. And, a lot of our samples that we have archived over the years are not fresh frozen. These are on paraffin embedded. It's kind of limits what you can do with them. And I think also what really kind of hurts is that they really have not been able to culture these cells. We don't have a big bank of just cell lines like we do for other, like, leukemias and solid tumors even. And again, there's no animal model as well that can kind of give us some clues as to what causes this disease to develop in the first place. So I kind of switched gears a little bit and kind of turns out to kind of where my aim is kind of coming from. Interestingly enough, there's been some rare cases of both an LCH and a TL cancer, kind of in the same patient simultaneously. And this one was kind of the most recent one in 2007. And so they actually found that both the LCH and the TL were actually clonally related, meaning they probably, they seem to think that they came from kind of the same developmental tree. And they actually found that they share the same activating Notch1 mutation, which is a mutation that we commonly see in TL, and I'll explain a little more about it. And so if you look at, in terms of the Notch signaling pathway, this is kind of an important pathway, unfortunately. Sorry guys, this is kind of dry. But this is like a really highly preserved pathway. It's kind of, you see it all the time in terms of development, and really has a big, big role in terms of hematopoiesis, excuse me. And I think just importantly, just before we go on, I just importantly want to just, in terms of signaling, you have ligand binds. This is the protein itself, and kind of the way it's activated is that there's a secretase here, an enzyme here that cleaves, and so you have like an intracellular portion. And that's what we're measuring in terms of I'll talk a little more about kind of some downstream measurements that are done on this. So it's just important to know that this is on a membrane, and then when it's active, it's actually in the cytoplasm versus attached to the membrane. And so these are, like I said, these big transmembrane proteins. They have a bunch of receptors. There's 4 of them, humans, Notch 1 through 4. And they also have different ligands. And for our case, I just wanted, we're going to talk a little more about the jagged ligands in terms of LCH62. So, this Notch 1 mutation is not, it's not like a big shock to see it in this T leukemia patient, because we actually do see it quite often in T-cell leukemias. Interestingly enough, the first one they found was just a big chromosomal translocation. This was in the 1990s. And then when they actually started doing more and more in-depth sequencing, they actually found that it's probably, but maybe even a little higher than 60% in all T-cell leukemias, all ages. And again, these are big. This is a big developmental pathway. So the downstream targets of this Notch syndrome system are guys that are actors that we see in all sorts of different cancers. So this is not, this is not terribly shocking. This could be an important oncogene in terms of other hematopoietic systems, since it has such a big regulatory control over all these important cell-stimulated target genes. So. Based on this, another group decided to look specifically at LCH, notwithstanding that single index case of the TL and the Notch mutation one. And so this was, unfortunately, the study looked mostly at single system LCH. So, bear that in mind, these may be different, kind of ways that these disease presents, but they found, they first did a comparative gene expression array again, and they actually, on these, on their LCH cells, and they actually noted that there was an upregulation of this JAK2, which is the jacket, excuse me, the ligand that binds to Notch. And so, lo and behold, when they went back and started looking more specifically at Notch, they, here's the Western blot here, which showed, this is an intracellular part, Notch IC. This is, in theory, should be an active Notch that's actively causing downstream effectors, but as you can see, these are some biopsies that are just ripping Notch. And these are positive control jerkat is a T cell line that we know has active Notch signaling. And then they did some staining, which was really pretty, so I put it here. So Langerin, this is CD207, so we should see, you expect to see it on the marker. And here's Notch, and so if you look at overlaid, it's intracellular. Again, this is IC. And it's the same thing over there, with just, but that's with CD1A, which is the other surface marker that we use for Langerhans cell histiocytosis. So I think what really was exciting about this paper is, and they then took CD14 cells, which are in theory myeloid precursor cells from just peripheral blood, and then they cultured it with some other TGF-beta, GM-CSF, just try to induce kind of differentiation. But then they also put it on a layer of cells that were expressing this JAG2, of the jagged ligand. So basically they took these cells and they just kind of turned on Notch signaling these cells. And what they actually got, when they looked at the cells that came out of that, they actually got cells that looked like a Langerhans cell histiocytosis marker, meaning if you look, so I think, let's see, this is the one that really we should look at here. They tried different permutations. So you want one that has a big Langerhans cell, Langerhans positivity. So not much, not much, not much. And they get two big spikes over here. And then if you look at, they also looked at E-cadherin, which tends to be on dendritic cells. And so what they were, their big point with this is that Langerhans cells have Langerhans with CD207, but they do not have E-cadherin. And so if you look at, they use the IL-4, which is an interleukin. You see you have Langerhans positive, E-cadherin positive. And you say, well, that looks like a more of a mature dendritic cell. Versus when they do this jagged or notch signaling pathway, you see a big spike in Langerhans here, but still remain negative to E-cadherin. So they say, well, this at least gives you a phenotype that looks like an LCH cell. Whether or not this is a good model or not, who knows? But this at least gives you something that looks like a good phenotype, which is kind of interesting because this would be the first kind of cell cultural model for this disease. So I guess in terms of what I want to do with this, and I'm mostly interested in the WIP pathway, which I'll give you another pathway. This is a beta-catenin, which again I'm sure we've at least heard of. I think we've talked about it briefly. And malignancies and whatever the last 30 minutes. So our kind of big hypothesis here is that we think that WIP signaling is at least playing a role in LCH pathogenesis, and I'll explain to you why. First of all, no one's really ever looked at WIP signaling in LCH specifically. There is some evidence that WIP may be important in terms of Langerhans cell development, but not necessarily LCH. There have been a few studies looking at like epidermal Langerhans cell where if you do knockouts, they don't develop well unless you have some activation of WIP signaling. So their kind of big takeaway point from that was that this is a pathway that's occurring downstream of the notch pathway. So what is WIP? This is another huge family of signaling family. And again, this is a big signaling pathway in terms of hematopoiesis. So there's a couple of different ways you can signal through WIP, and I think we're focusing mostly on the canonical WIP signaling. And this is really just primarily cell determination. And I think it's important to think that really what's causing the activity of this WIP signaling is this, and what we're really interested in measuring is hypophosphorylated beta-catenin, which is in the nucleus of cells, and that's going to cause big different transcription factors. Interesting enough, again, we see a lot of T-cell lymphomas again that seem to have positive WIP signaling here. So here on this percentage here. So the B cell neoplasms, not too much here, but lymphoblastic T cells seem to have a fair amount, even mature as well. Like I said, here's the WIP thing. It binds. They have awesome names. These are all derived from fly models. So they're all like winglets, binding to frizzle, just depending on how the flies end up looking. So it's kind of interesting. But basically, when you bind, the WIP binds to these like big receptors, and there's a bunch of different frizzles. There's a bunch of different WIP bindings. But what you end up having is this beta-catenin here, and it's usually in complex, and it can't go anywhere when it's in complex, and then this signaling causes it to release from complex and go down here, where again you see MYC, CD44, CCND, like all these, and then MMP7. These are big genes that are involved both in cellular differentiation, cell growth, and as well as migration and adhesion. That's sort. So again, there's a lot of downstream effectors, kind of in some ways very similar to notch, but in some ways not. So so again, no one's looked at specifically in Langerhans cell histiocytosis. In terms of dendritic cells, there does seem to be a role, as I said previously. if you culture what you think are stem cells, you give them some growth factors, and then you just blast them with wincingly, you do seem to get a shift away towards this classical dendritic cell lineage. You get a shift towards that classical dendritic cell lineage, which we think is where the LCH cell is a little more closely related to than this other type of dendritic cells, which I'm not going to go into too much detail. And again, you also see a lot of crosstalk with Notch1, so these pathways seem to be very intimately related in terms of the signaling as well. But overall, what they think they see when you give at least myeloid progenitors beta-catenin is that you seem to get hematopoietic stem cell reconstitution, meaning there's like a bigger pool of stem cells, but that they are not necessarily more differentiated. It's kind of like the big takeaway. And there's, different cell types are going to respond differently, which is kind of the really confusing part of cell signaling. But so I think right now what we think is what's going on is that Notch here signaling, and it kind of upregulates this Wnt signaling. And so what the best thinking that we have right now is that this Wnt pathway may be acting downstream of the Notch pathway, and that maybe, and it's possible, well, at least I hope, that maybe those cells that may not have necessarily a bare Notch signaling may have signaling abnormalities in this pathway to make up for this or to drive pathological phenotypes. So I think what we're going to do in the lab is a lot of phosphoflow cytometry, which we've heard about it briefly, but really I think the advantage of the phosphoflow, and again you can't do this with paraffin samples, but we can do it with both our T-cell leukemia patient sample as well as this new cell culture that we hopefully are going to get underway here, is that it really does allow kind of a multiplex analysis of different signaling events. The premise, of course, is that the different phosphorylation status of these proteins could directly correspond to biological activity, which we know for beta-catenin at least is true in terms of like the hypophosphorylated one being considered the active form versus the versus the hyperphosphorylated. And it also is a nice technique in terms when you have really big mixed cell populations, because you can't look specifically at one cell population in this huge pool of gunk, as opposed to like Western blotting, where we have to rely more on like a more pure sample going in. So here kind of is like a beautiful picture I found in a review article on phosphor flow. And so really like the basic experimental protocol here is you take your cells and then you give it a stimulus, A or B, or A plus B. This could be stimulus, this could be drug treatment, this could be something to cause more phosphorylation, cause less, it could be anything. And then basically what you do, you have antibodies that are raised to the specific phosphoepitopes of the proteins itself. So for example, this purple valuable red thing only binds that. And if that phosphate isn't there, then it's not going to bind with a high affinity. And so at the end, when you put it on the machine and you read fluorescent intensity, so you allow these things to bind and then you wash away whatever is not bound. So then when you measure intensity, for example, this guy, where you have the antibody to this guy, this phosphoepitope, you just see this here, and it gives you like a readout in terms of median fluorescent intensity. And then like this B is like the opposite of A, where now you have an antibody raised to this green over here, or you can even do both. And the beauty of phosphoflow is that you can run multiple antibodies at the same time, depending on how many channels your machine will allow you to do. So you can get a readout of multiple things, which is super neat. And so again, this is kind of just, and this is a basic overview. You got your cells, you treat them, you fix and fertilize. You got the problem, of course, you need these antibodies to get inside the cell, so you do have to do some fixing and fertilization to them, and then you have your readout. And then I guess a couple couple of years ago. I spent a good amount of time working on some antibodies that are kind of important in this beta-catenin system, kind of verifying them to see if they work or not, which takes a little longer than you think. And I think just briefly talking, we used both the GSK and the anti-active beta-catenin, which is really hard to say. I don't never say it right. So this is the antibody that's raised very specifically to that hypophosphorylated beta-catenin. Yes, okay. I always, yeah. And then GSK is a protein that, let's see, I have a picture here somewhere. Is a protein that will be attached to this beta-catenin in the cytoplasm if it's not active. It's it's a big complex of proteins, but it's one of them. And so really, what we wanted to do with this verification is give these cells both drugs that would increase it and drugs that we think would decrease it, and hope that when we run it on the machine that we actually see some shifts. That's at least the hope. And so we did some PI3 kinase inhibition. I don't really want to go into that too much, but that basically you inhibit PI3 kinase, you get less AKT, you get higher levels of this GSK3 active protein, and then the end result is you get less beta-catenin, less active beta-catenin. Yes, I should say it that way. So that was one kind of test we did, and then we also used a direct Wnt agonist, which is just a ligand that binds to frizzles. And then again, we would expect to see, if we Wnt signaling, we're going to have less GSK3 that's actively bound to beta-catenin, meaning you're going to have higher levels of the active beta-catenin, which I hope makes sense, because for me that took like a month to figure that out. And so here, this is kind of just typical readouts that we get from the machine. And so I don't know if any of you have experience looking at flow data, but here we have just unstained. So you would expect it to be almost no signal, but you're going to get some background noise. So here's 0. So this is what we consider unstained, and then here is basal, unstimulated. This is using leukemia cell line. And so what I did here, we have different countries of the Wnt agonist. So again, we see higher levels of active beta-catenin. And then if you use PI3 kinase inhibition, you see decreased levels like we thought. So that was good to see. And then we did again with another cell line, which is not quite as pretty. But again, we see increased here. This one got kind of wonky. I think the cells were not happy with that level of because these are big cellular signals, so we are causing some effects on the cells by giving them these drugs. So, yeah. But the trend at least is there. You see a little increase here and then decrease there with PI3 kinase inhibition. This is just another T cell line as well. So briefly, because I want to leave time for questions. So what we are proposing to do. So again, we have this LCHT-all case that we have from we're collaborating with the guy down at Stanford, we actually saw this patient in the clinic there. And so again, what we're going to do is very similar to that first 2007 report. We're going to take it, we're going to look for these things clonal, do they have a Notch1 mutation? And then because we actually have frozen cells from this patient, we are going to try to look using these kind of phospho-flow techniques that we have developed to kind of look at that, just to see if there's also some beta-catenin upregulation. And then again, you could also use older kind of immunohistochemical methods to kind of verify that as well. Like, is this beta-catenin nucleus, is it bound still to the outside, and then it maybe not be active. And then the other big thing we're trying to do is get the cell culture thing up and running, which is it's getting there. Had some steps, but and so then basically we want to get some cells that look like a Langerhans cell and then look to see. And then if we have a cell culture, then we can use things like treat it with different drugs, different inhibitors, and try to see what kind of phenotypes we can use out of it. So those are kind of the big overarching designs. And yeah, I think that's it. So I'll take sorry again for all that sickness.